Okay. So species interactions. So this section of the course now we'll be getting into different interactions, biotic interactions, looking at how the microbes interact with other living organisms, including other microbes. And so this slide set is kind of a general concepts of how might these interactions work. And I'll be giving you some examples to try to explain some of this. But with this figure, they're looking at time on the x-axis and then the evolutionary events on the y-axis. And they're basically trying to show how, there's some big moments in the evolutionary process to think about. One of the main things to think about, though, is that microbes were here first, right? They were here first. And so anything else that evolved came into that world of microbes. And so, of course, microbes are going to profoundly influence other organisms that are evolving and you have a lot of interactions going on. So you can see some of the main big physiology going on that changed atmospheric conditions with CO2 fixation and nitrogen fixation. Ongoing, evolution of the biochemistry of the microbes, and then eventually you start getting into eukaryotic life. But, for many, several billion years, it was the microbes that were dominating and everything else came into their world. And so when you start thinking about how the microbes are interacting, they often use these types of terms. So on the top half, you have positive interactions. And you can see a range of different terms here. And then on the bottom, we have some negative interactions. Now, one thing I do have to say is that this can get there's a lot of gray area with some of this. So sometimes you might see commensalism, for example. What that means is one organism is benefited, the other one is neither benefited nor harmed. And that can kind of be, yeah, maybe, or maybe they're both benefiting. And so there's a little bit of gray on some of this. And so I just want to caution you that it's not this really hard black and white designation. There's some exceptions and some things to think about. So I guess take some of this with a grain of salt. And then I don't know why they put neutralism up there. That's kind of a moot point. But here you have symbiosis on the far right here. And here they're showing bilateral. That means it's a win-win for both interacting organisms. And then you can see that the co-evolutionary history of symbiosis is long. So what they're meaning there is that there's been a lot of time for these two organisms to form this association and for it to be a true win-win in this case. Parasitism also, you can see, has a long coevolutionary history. Sometimes you have such a impact on each other that the parasite, we've mentioned before that it can lose some of its own ability to live independently and will purge a lot of its genome and allow for the host to take over a lot of that physiology that it used to do. So on the other hand, short coevolutionary history can happen with competition, of course. And so just thinking about some of these terms, we're going to go through some of the interactions here. So let's start with some of the positive ideas and that commensalism, which again can be a little gray in terms of this, but basically when they think about it, they're thinking about the idea that one organism's benefited, the other one's not really affected. And so, for example, you might be able to say that with two microbes that are interacting. One is facultative and the other one's an obligate anaerobe. So if you're a facultative, that means if there's oxygen present, if you're smart, you're going to use it. So if you are hanging out with an obligate anaerobe and you use up all the oxygen, that's benefiting the obligate. All right, and so that's what they're going on with the idea that the obligate anaerobe benefits, the facultative is not really affected. It's just doing its own thing, using up the oxygen in the area, allowing the obligate to establish and hopefully thrive. So that's a classic example of commensalism. Other ideas that they've thrown out is that you have this idea of growth factors or vitamins that are being produced by one organism and excreted, and then a nearby organism can take that up and use it. So they've looked at, for example, this interaction. You don't have to memorize species names here. These are just ideas to get you thinking about the concept. I'm more concerned that you understand the concept, not memorizing who does what. But understanding that there can be excretions of growth factors that by one, and then a neighboring microbe can then use that to their benefit. There's also the idea of when you have a consortium or a group of microbes, you have microbes that are interacting nearby, and if they're excreting different enzymes and acids to break down compounds or rock material, for example, that the solubilization of that can help others nearby. So that's a commensalism concept as well. And then you have this idea of getting gaseous compounds created. So we talked a little bit about that with your Winogradsky column. So they often talk about like methane getting produced by a bacteria. And then of course that's a gas. So then that starts to get leached up or diffuses up into the water column. And then you have methane bacteria that use methane, oxidize it, strip electrons off of it. So that's a win for those types of bacteria. We talked about the hydrogen sulfide in particular, because in your Winogradsky column, you did give some sulfur material in there in the recipe. And so you can get this idea of basically, one microbe's waste is another microbe's use. They can take that and use it. Even methane synthesis, the creation of methane, methanogenesis. So again, this idea that you have, it's really, they don't think microbes live out in their natural life, in the natural habitats all by themselves. There's always organisms that are living in groups. And so some of the positive ideas here is that you do have waste from one being used by another. And then you have this idea of kind of a consortium of microbes that are acting to break down more complex material, whether it be rock material or cellulose, something like that, where you can have a group action. So again, sometimes, yeah, one partner's benefiting, the other one's neither harmed nor helped. Sometimes commensalism, I think, actually goes into more of a mutualism, whether it's a win-win. And so it's kind of a gray area, and it kind of depends on the example you provide, I suppose. This is a classic one that's often given as this idea of metabolic commensalism. So they call it co-metabolism. So in this example, they're showing that Mycobacterium is acting on propane. They're actually breaking propane down and stripping electrons off of that for their ATP needs. And you can see this dotted line, the cyclohexane. What they're trying to show here is that as Mycobacterium is doing its thing with propane, they often say it gratuitously breaks down cyclohexane, but it doesn't work with cyclohexane. But Pseudomonas does. And so if Pseudomonas is around, it's going to benefit from Mycobacterium. Again, yeah, okay, this is co-metabolism, does Mycobacterium truly not make any use of cyclohexane? They're claiming it doesn't, that it's just a gratuitous example of breaking down the cyclohexane that then the Pseudomonas can take advantage of. So there's these types of ideas where they're claiming the Mycobacterium isn't really benefited by that. It's just doing its thing, but the Pseudomonas is benefited. Here's another idea, removal of toxic substances in the environment by a group. If you have Beggiatoa, for example, remember hydrogen sulfide is toxic in high doses. It's toxic to any living organism. And so if you break down hydrogen sulfide, then other nearby microbes that are sensitive to it can survive and coexist. Again, Beggiatoa stripping electrons off of hydrogen sulfide is definitely benefiting because that's a good electron source. So, but anyways, they're saying that this is helping other nearby sensitive microbes to go ahead and survive in the presence of hydrogen sulfide. Heavy metals too, of course, too much of heavy metals, you're not going to do well, including microbes and other organisms. And so you have the breakdown or the precipitation. So if you can precipitate a heavy metal, essentially you make it more inert and less harmful. And so precipitating out heavy metals is a good idea. If you can't break down, I mean, metals can't really be broken down per se. They can be precipitated or sequestered. And then volatilization can be done. If you think about mercury, for example, can be volatilized. That is one that is capable of that. That's why it's so dangerous to work with. You probably never had to deal with thermometers that had mercury in them, I'm guessing, but maybe some of you have. But that was when let's see, when the Weston Hall was built, and before we could move in there, all of our mercury had to be gone. Like none of the thermometers that had mercury, anything that had mercury in it, we had to get rid of before we were allowed to move our labs into that building because of the new OSHA laws. But I have fond memories of chasing mercury around on the floor when a thermometer broke, trying to sweep it up and get rid of it. And that was back in the day when they didn't really know that you shouldn't be doing that. So anyways, mercury, yes, is volatilized. Got to be careful around mercury. Okay, here's an interesting example too. It's a kind of a difficult picture, but what you see, the bigger blobs there, that's algae. And all the tiny little blobs are bacteria that are hanging out on the algae. And what we have happening here is that the algae is, exuding off different compounds that the bacteria can take advantage of. And what they found is that it's almost, in my opinion, a little bit more synergistic, which they mention here, because it seems that the algae, when they do experiments, at least in the lab, the algae can't really grow if the bacteria aren't with them. So in this case, synergistic means that they're both benefiting from this interaction. And so it seems to me it'd be more of a synergistic than a commensalism. So I've seen this presented as both commensalism and synergistic. So I would lean towards synergistic on this, with the idea that it's not obligatory in the sense of these bacteria can do fine on their own. So the bacteria doesn't need the algae, although the algae technically, I guess, at least in the lab, when they ran the experiments, the algae did need the bacteria. And so sometimes they'll throw out these terms like proto-cooperation because they're not in a tight relationship through a coevolutionary process that's been going on and on and on. But when they do hang out together, they're both benefiting. Synergism also includes this concept syntrophism, which is just, again, when you see troph, it means to use or to eat, if you want to think of it that way. And so two or more populations are helping each other nutritionally. And when there's countless examples of this idea, again, where you have, again, the waste product of one being used by a nearby microbe. So here you have, so they have line graphs here of the growth of A, B, C here. So when B and C are if the lactobacillus and the enterococcus are grown all by themselves in minimal media. So minimal media means you just provide the bare basics, nothing complex. You don't throw in any complex media. It's all defined and very minimal. And so A or B and C is showing if you grow lactobacillus and enterococcus all by themselves, they don't do well in a minimal media. Grow them together and they really take off because the waste product, lactobacillus will make folate and excrete it. Enterococcus needs that. On the other hand, enterococcus makes phenylalanine. Lactobacillus needs that. And so if they're grown together, then they really take off. Grow them alone in minimal media and they don't make it because the minimal media doesn't provide the folate or the phenylalanine. And so lots of examples of this as well, especially when you do these experiments in the lab, you can demonstrate this type of idea pretty readily. Here's another example. So Anabaena is a cyanobacteria. It's got many, many vegetative cells that kind of string together like a string of pearls. And then one of those cells develops into a heterocyst, which is where they go ahead and do their nitrogen fixation. So in this heterocyst, it's a very special cell. It allows for the breakdown of dinitrogen to ammonia. Well, in order to do that, you need the dinitrogenase enzyme. This enzyme is really sensitive to oxygen. And so this becomes a problem because the cyanobacteria is doing photosynthesis, which is giving off oxygen. And so the heterocyst could be getting exposed to a lot of oxygen. So then the dinitrogenase falls apart and doesn't work. So that's not a good idea. So often what they find in more natural habitats is that there'll be other microbes hanging out with Anabaena, and they'll be preferentially hanging out right around the heterocyst. And when you, with Pseudomonas, again, if they can use oxygen, they will. That's a preferred electron sink. And so the Pseudomonas will start to gather and be in the area where there's oxygen being given off by the cyanobacteria, other nutrients that are getting sloughed off from the Anabaena. And in the process, it's lowering oxygen around the heterocyst and allowing for that enzyme to work optimally. And again, Pseudomonas would benefit for the enzyme working correctly because Pseudomonas can use ammonia too. And so it's definitely a win for the Pseudomonas if they hang out in that area, and it's a win for the Anabaena because they can keep the nitrogen fixation going. So this is a little grainy, but it's trying to show, I don't know if you can make out, these are like the vegetative cells of Anabaena. And then this dark blob is the heterocyst. And then I don't know if you can kind of see the dark blobs that are congregated. They're all in this area. And they're kind of hanging out preferentially around the heterocyst. And again, they are, have done some careful studies where the Anabaena is able to essentially feed the Pseudomonas, and then the Pseudomonas is grabbing the oxygen and keeping it low levels around the heterocyst. Okay. Another positive interaction that's often talked about, mutualism, where it's a definite win-win. Lichens are a classic example of that. Lichens are amazing. This interaction, once they form this relationship, it's stunning how different they look. You can grow them separately, but you put them together and they form this unique tissue-like organization and partnership. And so, technically, I guess you could say they're ectosymbionts in the sense that they're the partners aren't living within each other in the cells or anything like that, but you definitely have this tissue-like development. So this is a picture of a lichen under the microscope, and all the kind of darkish reddish blobs are the algal cells. And then all the green that you see through here, all that green is fungal hyphae. And so they're mixed together in this tissue-like organization that we call a lichen. And so when you look at this partnership, it's fungi hanging out with algae, or fungi hanging out with cyanobacteria, or all three hanging out together. And now they've discovered too that sometimes there's some interesting yeast species that are definitely part of this lichen mixture. And that solved actually some interesting conundrums they were having with some of these lichens behaving differently than expected. And then when they looked more carefully, they realized, oh, there's a yeast present and they're doing that unexpected thing. That was kind of confusing until they figured out the yeast were there. So again, this image is trying to show that you do have this kind of tissue-like organization. And sometimes you'll even have this is tissue that's kind of organized to kind of latch onto the substrate. And so you have parts that are, designed to latch firmly onto the side of a tree trunk or onto a rock. And then other parts of it are more upright off of the substrate. And then this area is a little confusing, I guess, because it's a bunch of red. And I just got done telling you that the algal cells are red. But that red stuff, that's not algal cells. Those are spores being produced by the fungus. And so they often, when they go into spore production, they typically will form it up there like that on the surface, which kind of makes sense because they want those spores to get released. So there's raindrops or something stepping on it that will help release those spores. So tissue-like organization often. It's still kind of a mystery on how they form this relationship. No one has witnessed a lichen forming. That'd be a good master-student project. Go sit in the woods and watch for lichen to form. Grow old and starve to death out there. So no one's witnessed it. And so and I'll explain a little bit about how they actually colonize new areas, but here you're seeing this idea up close where you can see the fungal hyphae a little bit better, the green, and then you can see the algae interspersed. And then again, all of that top portion is fungal spores. And closer still. So all of those are clusters of spores. Those are called ascospores, and the sac of tissue is called asci. And so the ascospores are inside all those asci. And then the yeast I was telling you about, that was discovered by scientists in Purdue, where they were looking at this kind of confusing lichen behavior, and they were able to figure out that it was actually a yeast involved in it. So they're starting to look for that more carefully with other lichens, and they're starting to see that that's kind of a common thing, that there'll be some yeast showing up. This finding could explain why many genetically similar lichens present have wildly different physical features, and why scientists have been unable to synthesize lichens in the lab. So yeah, they haven't been able to force a lichen relationship. They can't get it to happen, and they've not witnessed it in the wild either, so. Kind of still a mysterious interaction. So mutualism, in the sense that the photosynthetic organisms are definitely providing carbohydrates to the fungus, which of course the fungus needs. And then the fungus seems to provide a lot of protection because the algae and cyanobacteria are kind of mixed in with the hyphae and that tissue-like organizations. The fungus is really good at breaking down rock material and solubilizing minerals off of that, giving growth factors to the photosynthetic organisms. And so it's definitely a win-win. When they look at most lichens, it's these three genera that seem to be the algae that are hanging out. So I would like you to know these three. It's only three, but Trabuxia, Pseudotrabuxio, and Trentopolia are the three that are dominating most lichens, not all. And I'd like you to know this too, please. The nostoc is the cyanobacteria that seems to dominate most lichen relationships. So they estimate that about 90% of all lichens have one of those four involved in the lichen relationship. Most of the fungi involved are ascomycetes, that group of fungi. There's several different groups of fungi classes, and ascomycetes are dominating. There are a few basidiomycetes. And then don't forget, the yeast belong into the basidiomycete category. So that's why they say, yes, basidiomycetes can be present. Zygomycetes, there's some evidence that they're there, but there's still a question mark on that idea. So by far, the ascomycetes dominate. So I would like you to know that. They think that these processes have been separate over evolutionary history. But once formed, it's definitely a partnership that can benefit both partners. When you look at the scientific literature, they name the lichen relationship by the fungus. So they'll just use the fungal species for that. Under the microscope, you might see something like this when you take some of the tissue and look at it. So you can see the fungal hyphae. And then the brown are the cyanobacteria or the algae. Here, you're looking at the lichen body. By the way, they often call it a thallus. That's the fancy word for the lichen body. They'll call it a thallus, Thallus. So you might see that in the literature, but basically you can see how there's like different regions. So that's what they mean by like a tissue-like organization. These are not roots, of course, it's not a plant, but they do have structures that allow for attachment. And then here, I would like you to know this term, please. It's not gonna let me, Soredia. So this is how lichens will colonize new habitat. So instead of trying to find each other all over again and establish a new lichen, partners break off together and go nearby. So Soredia is the fungus and the algae or cyanobacteria in a group that breaks off. And so usually what you see is the algae or the cyanobacteria is wrapped with fungal hyphae. And it's usually at the surface of the lichen like that. And you can imagine like a water drop or a deer walking across that would break the Cerediia free. And then they can tumble into a new habitat and hopefully set up camp together. And so they go together as a unit to colonize new areas. So B is just looking at Cerediia up close. Here's a better picture of that. So you can see the fungal hyphae wrapped around. So that solves the problem of being able to find each other. Once the lichens form, they can keep colonizing if they go out in a unit together. So if you look down on a patch of lichen, often that kind of granular look to it, that's Cerediia. And so you can't always see it. And sometimes you would need a hand lens in order to really look at it more carefully. But some lichens, you can see the Cerediia forming on the surface. The other thing to be aware of is lichens are incredible at hanging out in habitats that are very inhospitable to us. Here, this is, I believe, yeah, Antarctica. And you're looking at cliffs and mountains on figure A. Figure B is showing a close-up of some of the rock material. And you can see this discoloration toward the surface of the rock. That's lichens. Those are lichen hanging out together in the rock. And so we'll have the word there. So the fancy word for that is endolithic. Endolithic, which means within the rock. So negative 60 degrees Celsius in the winter. So they just tuck down into the rock material. They're still able to access sunlight, but they're protected in the rock. So pretty amazing abilities. Lichens can handle all kinds of adverse conditions. And, you know, if you've ever noticed the lichens, for example, on sugarloaf or something, or hogback, you can see that, you know, very often they look really desiccated, and if you walk on it, they kind of crunch under your foot, and you might think that they're dead, but they're not. They're dormant. And if you go during a rainstorm, you can see that they do, often they'll green up before your eyes. They'll start to turn green. You have to wait a while, but sometimes you'll see that happening. And they become active again. So lichens are also commonly thought of as indicator species for pollution, because they are very sensitive to pollution. They have no way to excrete it out of the tissue, and so they kind of sequester it. And over time, it can really build up in the lichen body. So here, this is a classic example over in England where they were tracking presence of lichens in the city center, which is 0.0 is the city center, and then miles away from the city center. And you can see that it took a while before lichens started showing up. So demonstrating that the center of the city had enough pollution where lichens were not happy and weren't able to really persist. And then as you get further away, then they started showing up. So if you've noticed here in Marquette, have you noticed lichens in Marquette? Start looking for them. Even walking downtown, I can see them. And they're everywhere. And so that's really kind of reassuring that our air pollution isn't so bad. There's lichens all over the break wall, all that orange stuff that you see on the break walls, that's lichen. And so they're everywhere here. And I find that really reassuring because air pollution really can do a number on them. That's the one thing they're super sensitive to, especially sulfur and containing compounds. They're beautiful, many different forms of their body, the thallus, lots of different colors. The different colors are pigments that help them deal with too much sunlight energy, because very often they're on bare rock or, you know, on the sides, you can find them on rooftops, you can find them on gravestones, cliffs, tree trunks. They're all over the place, even, you know, certainly on the ground too. But that extra pigmentation helps protect them from too much sunlight. So this is the one, Xanthoria elegans is the one found on the break wall. Beautiful orange. One of Dr. Robertus's students, Ryan Rutherford, he did a whole master's thesis on lichen communities in the Huron Mountains. And so I think he even gave I wonder if I can get my hands on that. He gave a talk. There's a TV show where he gave a talk on lichens in the area here. I'll see if I can find that and post it. So lichens are a fascinating interaction. One thing to be aware of is, if you can avoid it, try not to walk on them, because they don't grow very quickly. They have a very slow growth process, I guess. Also, if you take any out of a habitat, be very stingy about that, because you don't want to, you know, take a whole chunk out of an area. They just grow so slowly. Dr. Robertus and I went on a workshop in the UP looking at lichens of the UP several years ago, and there was like 30 of us there. And so we had, you know, the workshop presenter was very careful to tell us, you know, be careful where you walk, only take tiny samples, you know, so that we could take them and go back and look at them under the microscope and study them more carefully. So just respect that they are a slow growing organism, a collection of organisms. But they're incredibly hardy and are, except for the pollution idea, they struggle with that. So the lichens aren't really, they're called ectosymbionts because they're not growing within each other in any way. Here's an example of an endo instead of ecto. So this is a protozoa with algae inside. And so the actual cell that you're looking at here is paramecium, which is a eukaryotic cell. It's a protozoa. And then chlorella is the algae. And you can see that it's chock full of chlorella. And what's really cool about this is that from the algal perspective, the paramecium is definitely giving protection. It's physically inside the paramecium. The paramecium has flagella, and so it can move around. So that's great for the chlorella, because then it hopefully will get to areas of sunshine and can access sun for photosynthesis. So it can get out of shaded areas with the help of the paramecium. The paramecium is doing respiration, which provides CO2. So then, of course, that's good for the chlorella because it's doing photosynthesis, taking in CO2, fixing it into carbohydrates. And the paramecium also gives growth vectors. So it's this mobile unit that the algae gets to hang out inside of, gets access to sunlight routinely because it can be moved around. And then the algae, of course, is providing carbon through photosynthesis, oxygen through photosynthesis. So from the paramecium's perspective, the paramecium can actually get into areas that are devoid of oxygen, at least for short periods. And because it's carrying its own, you know, oxygen producing organisms with it. And so that's kind of nice. The paramecium can go and get access to food in anaerobic environments. So kind of a nice win-win there. The only downside is, at least experimentally, when they work with these in the lab, if you stress them and provide, like, usually what they do is they provide like a toxic chemical or something, or maybe lack of some kind of core nutrient that the paramecium needs, then the paramecium can turn on the algae and start digesting it. So they're using them for, give me some carbs and oxygen, but if I have to, I'll eat you, so in that respect, the algae is a little bit at risk, but in general, that's under a lab situation. Hopefully that doesn't happen too much in real life. Okay, we'll stop there because the next one is a little bit more than 2 minutes worth. So we'll finish this up and then we'll start getting into more specific interactions with microbes.